Okay, let's dive right into this explainer. Today, we're looking at a completely groundbreaking 2024 study that tackles literally every implantologist's biggest hurdle, and that is unpredictable bone resorption. If you've ever dealt with an implant failing because of chronic inflammation, well, you know exactly what's at stake here.
So this rigorously peer-reviewed paper is titled Bone Regeneration Revolution. It was just published in the prestigious International Journal of Nanomedicine, and it investigates the exact cellular mechanisms that cause those frustrating implant failures and, way more importantly, how we can actually stop them in their tracks.
But how do we actually do that? To answer this, we really need to move past all the heavy jargon and focus on how to stop the specific cells that are quite literally eating away your patient's bone during periimplantitis. We're talking, of course, about osteoclasts and the inflammatory macrophages that trigger the whole process.
Part 1. Research Question and Methodology. Testing Miniaturized PEMF Devices.
The researchers set up a really ingenious in vitro study here. First, they cultured macrophages and osteoclasts, intentionally activating them with inflammatory stimuli to simulate a failing implant environment. Then, they exposed these bone-degrading cells to PEMF, that stands for Pulsed Electromagnetic Fields. And get this, they didn't just use some massive, clunky lab equipment. They used miniaturized Magdent healing abutments, placing them right there in the petri dishes to emit the PEMF continuously for 30 days.
Moving right along to Part 2, Key Findings and Data, the hard evidence on macrophage polarization.
The RNA sequencing data reveals a massive biological shift. The data actually proves that PEMF significantly downregulates those pro-inflammatory M1 genes like IRAK3 and IRF8 by a huge margin. And at the exact same time, it upregulates the anti-inflammatory healing M2 genes like EGR2 and IL4R by a really remarkable factor. This isn't just some minor fluctuation we're looking at. The magnetic field is actively reprogramming the immune response right at the cellular level.
So the crucial point here is what those M2 macrophages actually do next. Under PEMF stimulation, these healing M2 macrophages release extracellular vesicles, or EVs. You can kind of think of these nanometer-sized exosomes as little biological text messages, and they act as a direct stop signal to the bone-eating cells.
And the impact of those biological text messages? It's completely profound. Under normal inflammatory stimulation, bone-destroying osteoclast genes like CTSK, NFATC1, and TRAP just spike dramatically. But look what happens when you add PEMF to the mix. That osteoclast lytic activity is massively suppressed. You are quite literally shutting down the exact mechanism that destroys your patient's bone, tipping the scale entirely in favor of regeneration.
Okay, Part 3. Clinical takeaways for dentists, moving from the petri dish directly to the patient.
Ultimately, what this means for you as a dentist is a fundamental shift in patient care. Let's be honest, without PEMF, you are so often stuck fighting chronic inflammation and unpredictable bone levels. But by actively driving that M2 macrophage polarization with PEMF, you shift your clinical outcomes from those unpredictable inflammatory risks straight into highly predictable, preventive dental care. You're instructing the patient's own biology to build bone rather than break it down. It's an absolute game changer.
And finally, Part 4. Next steps and enhancing implant predictability.
The science here is just incredibly clear, and the clinical application is totally ready for your chairside. Integrating this evidence-based MED technology into your workflow puts the control back in your hands. So make sure you follow Magdent on Instagram and LinkedIn at Magdent MED, and definitely visit magdentmed.com to book a demo. Just scan the QR code on your screen right now to get started. I mean, after seeing how we can literally reprogram cells to save bone, why would you ever rely entirely on luck when you can rely on cellular science?